So let's start by reviewing your morning routine. Most of us, in some particular order, probably got out of bed and dressed ourselves, brushed our teeth, sent text messages, walked to the car, and drove to work. And these seemingly simple tasks require an enormous amount of coordination and communication between the brain and the body. And for a person who has a condition called Friedreich's ataxia, this coordination diminishes over time, making these tasks that we take for granted increasingly difficult until they're impossible. Symptoms of Friedreich's ataxia show up during puberty and are relentlessly progressive. Patients usually need a wheelchair by the time they're 20, and most patients have a serious heart condition that leads to heart failure in their 40s. Most importantly, there is no treatment for Friedreich's ataxia. So our lab is doing research to help find one. Behind me, you'll see my artistic rendition of a few brain cells that we'll use to learn more about this disease. All of the cells in our body contain a molecular instruction book called the genome. And these genomes are made up of 20,000 units called genes, each of which is a recipe that makes a protein that our cells need. And just like our cells have 20,000 genes, these cells have 20,000 apps. And today, I've opened the frataxin gene app, because we know that a typo in this particular gene is the cause of Friedreich's ataxia. Frataxin is important for providing energy to brain cells, kind of like keeping a phone battery charged. But in Friedreich's ataxia, there's a typo in the frataxin gene that causes it to not work properly. The gene, or the app, is essentially switched off, causing the battery to drain until the brain cell dies. So the goal of my thesis project is to figure out what is switching the frataxin gene off so we can find ways to turn it back on. So we started this project by scanning the entire frataxin gene, looking at all the different parts to learn more about how it functions, kind of like scrolling through the settings on an app. And when we studied these settings in 75 patients in our lab, we found a new one that hasn't been studied before in Friedreich's ataxia. It's called DNA methylation, and I'll show you the results here. DNA methylation is a molecular off signal for genes that works like a volume control. And if the volume gets too high, the gene typically switches off. So when we measured this off signal, we found that the volume was quite low in the healthy cell, only about 6%. But the volume level of this off signal in the Friedreich's ataxia cell is turned almost all the way up to 100%. We also learned that the exact volume level is slightly different for each patient, and the higher the volume, the more severe the symptoms are. So we next wanted to see if we could turn down the volume in patient cells in the lab using different molecules and gene editing techniques, and it worked. We're actually able to turn it down to around 12% in these cells. And for the last part of my thesis, we're testing these cells to see if the batteries are recharging and to see if we can find a treatment for this disease. Thank you.